Okay, so let me give it a couple seconds more and then we'll start. Okay, so thank you everybody for coming today to today's seminar. And I'd like to just do a brief introduction of our seminar speaker. Okay. Today's seminar speaker is Dr. Erica Pratt. And as a brief intro, she is currently an NIH T32 fellow at the University of Minnesota, and she's in the laboratory where she's working in the laboratory of Professor Lori Parker. She received her bachelor's in mechanical engineering and biomedical engineering at Carnegie Mellon, and then moved on to receive her PhD at Cornell University with Professor Brian Kirby. She also was an NSF grad fellow during her grad career, where she focused on building microfluidic devices for capturing circulating tumor cells from whole blood. Since then, she's moved on to work on molecular profiling of tumors, both at the MD Anderson Cancer Center, as well as where her current position is at the University of Minnesota. And I believe she's going to cover quite a bit of this research area. The title of her talk today is Engineering Blood-Based Assays for Cancer. So I'll turn it over to Erica.
Thank you so much for the kind introduction and the invitation to speak today. Just a point of clarification before I get started, the right-hand side of my slides is intentionally left free so that you can auto-dock your video without worrying that it's going to block any of my data as I move through my talk. So I appreciate the opportunity to present my work and my future research program. Today I'll be discussing strategies to design blood-based bioassays with the ultimate goal of providing a suite of non-invasive clinical biomarkers that can be used to inform personalized medicine. So let's start with the basic question of what I mean by personalized medicine and why we need to build these tools. This is an area that has gained a lot of momentum recently. The goal of this strategy is to characterize a patient's tumor biology, usually through sequencing, which is what this image is showing here, where patients with different biological phenotypes are shown in different colors, and we're profiling them to try to stratify them to a therapy. Once these patients have been profiled, we attempt to match patients with known drugs that target the genes being altered or pathways encompassing that gene. The premise of the strategy is that we can improve outcomes by tailoring the therapy to each patient's unique tumor biology rather than applying a uniform treatment strategy to a population that we know is inherently heterogeneous. So a great example of that is something that was published in Lancet Oncology by the Pancreatic Cancer Action Network, where they've run something called the Know Your Tumor program, where patients around the country can call in, be matched with a clinical industrial partner to have their tumor sequenced. And then based on the genetic alterations that are detected, they're matched with a known drug that either targets that alteration or, again, a pathway that encompasses that gene. And the consequences of that are fairly dramatic, which is what's shown in this Kaplan-Meier curve, where on the y-axis, we're tracking patient survival over the course of time, which is the x-axis, where each individual hash mark represents a single patient. And you can see fairly dramatically that the red line, which are patients who underwent sequencing and were matched to an FDA-approved drug, had significantly improved survival outcomes compared to patients where no marker was detected at all, or for which their personal tumor biology could not be matched to a drug. Now, if you quantify this, using this matched therapy strategy, median survival was increased by over 95%. And that translated to an increase of survival of 1.5 years to almost 3 years, which for pancreatic cancer is an enormous improvement because the 5-year survival rate for pancreatic cancer is already under 7%. So this is a dramatic improvement just by stratifying patients to an appropriate therapy. So since this strategy works so well, why do we need non-invasive biomarker tools? Well, one drawback to current tissue biomarker profiling technologies is that the actual biopsy step itself can be challenging, laborious, or dangerous to the patient. And this often results in the number of samples that do not have sufficient material to be sequenced or cannot find a biomarker at all. And an example of that is what we're showing here now in non-small cell lung cancer, where 30% of patients who had their tumor tissue sequenced were able to be assigned to a targeted therapy. A biomarker was detected. However, for almost 70% of the thousands of patients that were tested, we found no biomarker at all. And the key question here is this a biological phenomena? There were 70% of those patients actually negative for a biomarker that could be actionable through a drug, or do we simply just not have the tools to be able to detect those biomarkers at all? In order to address this, the critical question is, do we have an alternative source of tumor information that has equivalent predictive power to a traditional biopsy? And this is something that motivates my work specifically and something we'll be focusing on for the duration of my talk. So liquid biopsies are a non-invasive method to obtain real-time tumor information. And let me define what I mean by liquid biopsies. That comprises basically any circulating tumor analyte that can be found in the body fluid, usually the blood, but also urine and saliva. There are several different types of circulating analytes that can be found, but for this presentation, I'm going to be focusing on two. So let's establish some baseline terminology. I'll be talking about circulating tumor cells, or CTCs. These are, as the name implies, intact cancer cells that are shed or have extravasated from the primary tumor, or circulating tumor DNA, which is heavily fragmented free DNA that has made its way into circulation. Both of these biomarkers are extremely rare, and there's a strong wild type or normal background that can dilute the signal for both of these analytes. So in the case of circulating tumor cells, the contaminating background signal comes from white blood cells, which are the other major nucleated hematological cell in the bloodstream. So for example, in 1 ml of blood, there might be only one circulating tumor cell for every billion white blood cells. So that's a very low signal that you're trying to detect, and that requires careful engineering strategies and tools. Similarly, for circulating tumor DNA, there is a strong contaminating background from cell-free DNA. Within your blood, there is actually a lot of fragmented DNA that is circulating around through normal cellular turnover of your organs. And this is perfectly normal. There are no mutations associated with cancer. And this, again, drives the frequency of our target of interest quite low. As little as 0.01% of the entire cell-free DNA population will contain a tumor DNA fragment. So we really need careful strategies to be able to detect these alternative tumor surrogates so that we can use them instead of a traditional tumor biopsy. One of the first questions you might have is, well, do these biomarkers actually provide any diagnostic value? So if we go back to that data that I showed you a slide ago, where again, 30% of patients were positive for a tissue biomarker, but 70% were not. If you simply go back to those same patients and now test their blood, we're able to increase the number of detected mutations by 65%. So we captured a large population of patients that actually were positive for a biomarker that could be matched to a therapy. It's just that their tissue specimen did not have high enough quality for us to be able to detect it. And most importantly, patients that were assigned a therapy by looking at liquid biopsy alone had indistinguishable outcomes from standard biopsy. This means that liquid biopsy gives you just as good information in personalized medicine as a standard tissue biopsy, but in a non-invasive and a continuous monitoring manner. The reason this is so impactful is because liquid biopsies can now be integrated into every stage of cancer treatment, whether there's a detectable tumor mass or not. And that's especially meaningful in early stage detection, where you might not be able to see a tumor on CT scan or standard clinical diagnostic at all. Or, for example, a patient that's already undergone surgery, where we think they're technically cured, but there might be cancer cells that are disseminating. At each stage of cancer treatment, liquid biopsy can provide us a continuous and non-invasive insight that would otherwise not be possible if we looked at tissue specimens alone. So my laboratory and research that I'll be presenting with you today is really focused on integrating engineering and omics to develop non-invasive tools for cancer diagnosis and monitoring. I've developed skills in microfluidic device design and assay benchmarking, and I view all of my research through this lens of how can we find a technological gap and fill it to bring liquid biopsy to the fore to reach its full potential as a non-invasive biomarker. Liquid biopsy has incredible potential to revolutionize standard of care, but there are multiple areas where there's an engineering challenge to make these biomarkers clinically impactful. And in my presentation today, I'm going to share 3 cases where we were able to close a technology gap, which enabled us to ask a meaningful question about cancer treatment, cancer progression, or cancer biology. And the methods that we use to do that are 1, macrofluidics, and the second case, we use a genomic molecular detection strategy. And then I'll conclude with some proteomic tools that I've been building in my current research position at the University of Minnesota. But to start, we're going to focus on microfluidic technologies that allow us to close that engineering gap and detect circulating tumor cells. So to reiterate, liquid biopsy, it has high potential to replace tissue biomarkers, but the one key limiting challenge is how rare that signal is. So again, circulating tumor cells have to compete directly with white blood cells in terms of signal that we're trying to pick up. And again, there can be as few as 1 CTC for every billion white blood cells. So there are a variety of engineering strategies that people have employed to try to isolate these circulating tumor cells. And some of them are listed here. This is an older figure from back in 2008, but it's actually a really great reflection of the field when I started my PhD at Cornell. So you can see that each of these technologies are really focused on isolating circulating tumor cells based on a unique feature. So whether that be exploiting differences in cell size between the CTCs and white blood cells, exploiting differences in cell density between CTCs and white blood cells, surface marker proteins, RNA, DNA expression, etc. When I started my PhD at Cornell, most technologies were focused on exploiting a singular unique feature to isolate circulating tumor cells from contaminating white blood cells. And this certainly provided adequate sensitivity and specificity for us to ask a lot of interesting questions related to tumor burden and cells that we find in the circulation. However, when I started my PhD, one of our key questions was, we know that there are several physical differences between CTCs and white blood cells, and is there a way for us to integrate them? So the first topic that I'll discuss in the very beginning of my PhD was to address the question of, can we develop a combinatorial microfluidic strategy that exploits the multiple unique differences that we know exist in CTC phenotype compared to white blood cells? So the solution that I developed during my PhD to address this is something we call geometrically enhanced differential immunocapture, or GEDI. This integrated 2 different unique aspects. One is an immunocapture-based system where we're exploiting differences in surface protein chemistry, and also exploiting differences in cell size. So the theory behind GEDI is a microfluidic device that's filled with silicon micropillars, which in this schematic we're viewing from the top down with flow going from left to right. We have 2 different particles which are meant to mimic the size of a circulating tumor cell, which is slightly larger, and a normal blood cell, which is slightly smaller. As the circulating tumor cell travels through the device, it will eventually collide with one of these silicon micropillars. It will then be shifted up to a streamline that ensures that it continues to collide as it travels throughout the device, having the chance to have about 2,000 immunocapture interaction opportunities. In contrast, a smaller red blood cell, for example, might collide occasionally, but it will not get shifted onto a streamline that ensures this continued collision trajectory. So this method exploits the differences in cell size that we assumed at the time, which was based primarily on cell line data that suggested that circulating tumor cells were significantly larger than other blood cells. We, of course, now know, thanks to more sensitive technologies, that the range in the CTC size is much broader than what I'm showing here. But at the time, this is what we thought the median cell size would be, and we designed our parameters around that. So these 2 images at the bottom are showing you a real-time image of what these micropillars look like. So this is a zoomed out image of a glass version of our JEDI microchip. So this is, again, you're looking at the pillar from the top down, and then on the side, you can see that there's a circulating tumor cell that's been successfully captured on the side of this post, which is covered with a high avidity antibody. This is another image of our silicon microchip where, again, you can see the edge of the silicon micropillar and then circulating tumor cells that have been successfully captured onto this immunocapture coated micropillar array. One of the first questions that we had was, now that we're combining these two unique CTC phenotypes, we're exploiting surface marker differences and cell size, does this result in any net improvement in isolation compared to other microfluidic technologies? Obviously, we found that it did. Our capture efficiency and purities using this combinatorial method were about 40% higher than using similar strategies, which is what I'm showing in this series of graphs on your right. On the y-axis in the set of graphs, we have capture efficiency. So how many cancer cells did we capture versus how many we spiked in? And this is in two different solutions, PBS, which is a saline solution, versus whole blood, which is much more complex and difficult to isolate cells from. And then on this side, we have capture purity, so you can see how efficient our isolation and specific our isolation was. Since our CTC isolation was so efficient, we went forward with testing this in metastatic castrate-resistant prostate cancer patients, where we were able to detect circulating tumor cells in over 90% of samples that we assayed. And furthermore, we found that we could detect 200 to 400-fold more circulating tumor cells per ml than Veridix cell search, which is to date the only FDA-approved method to isolate circulating tumor cells. So this is already a categorical leap forward in CTC isolation technology. And that's data that I'm showing in this plot on the bottom, where we have CTCs per ml on the y-axis versus 19 different patient samples that we looked at. And the red bars are the number of circulating tumor cells that we isolated using JEDI versus the green bars, which are the number of circulating tumor cells from the same patient and the same sample, but run using the FDA-approved device. And you can see that there is a dramatic difference in efficiency of capture. During the course of my PhD, I worked on adapting the JEDI platform for a variety of downstream applications. I won't talk about all of them here, but I want to give you a general flavor of what those are. One major alteration that I worked on were different post geometries so that we could optimize our device for different target cells and different target particles. I also worked on modifying the base substrate of the JEDI microdevice. Our JEDI microdevices were and still are primarily made of silicon, but I had made optically transmissive versions that we could use in confocal microscopy and other imaging modalities. And I also looked on different bioconjugation strategies so that not only could we isolate circulating tumor cells, but that we could release them in a straightforward manner for downstream biomechanical analysis. And this is something that I received grant funding from at Cornell as an independent investigator during the course of my PhD. So over the years, the JEDI platform has been used to answer a range of cancer biology-driven hypotheses. I won't have time to go into all of them here, but I want to give you a general flavor and also point out the longevity of this technology. So I developed the JEDI microdevice back in 2008, and we're still using it up to 2018, and it's exact same configuration as when I made the CAD file in my first year of grad school, which is something that I find entertaining and gratifying. So in 2010, we did basic JEDI validation. In 2012, we were able to show that on JEDI microdevices, you can actually perform in situ chemoresistance testing. So using a patient's own circulating tumor cells, we can test them for chemoresistance and then stratify them to a therapy based on this response of those circulating tumor cells. We've also looked at single cell genomic profiling. We've applied this to precancerous and pancreas cysts. This technology ultimately, due to this publication in 2012, initiated a phase 2 clinical trial where circulating tumor cells were used as a secondary endpoint to decide whether patients should continue on their current course of therapy or switch to another form of therapy. We've also used this in a variety of pancreatic cancer studies and also to even study the effects of exercise on colon cancers, CTCs. So I haven't been involved in all of these studies. The ones that I am an author on are highlighted in blue. I'm going to focus on one to give you a flavor for the types of downstream assays we can perform with this technology. And the one I'm going to focus on is single cell genomic profiling. So we focus on single nucleus sequencing for a variety of reasons. One, we know that cancer is inherently heterogeneous, so looking at the single cell level will give us a lot of information that would be lost if you do bulk or averaged measurements. We looked at copy number variation, which is the gain or loss of whole sections of a chromosome, which is what's shown in this diagram here. We know that this is correlated with patient mortality. And importantly, it requires very sparse sequencing coverage. So you can use a single cell to measure copy number variation relatively straightforwardly compared to trying to do, for example, whole genome sequencing or whole exome sequencing. An example of what a copy number variation profile looks like is shown at the bottom. So on the y-axis, we have copy number, and then on the x-axis, we have absolute position, where we're moving from chromosome 1 to the sex chromosome on X and Y. So if this were a normal cell, you would have 2 copies of every gene. So it'd be a straight line across at 2 until you get to the sex chromosomes X and Y. However, this is a cancer cell, so as you can see, there is a lot of dysregulation happening here. The lines are moving up and down. In some sections of the chromosome, you have up to 5 copies. In some sections, you have less than 2. For this type of cell line, you have a hypertetraploid type signature, so the baseline is at around 4. This is a copy number variation profile for about 150 cells. For single nucleus sequencing, the current state of the art at the time and also to this day is upstream pre-enrichment with FACS or flow cytometry. There are a few drawbacks with flow cytometry for single cell isolation. One is that it's very lossy. So if you have a rare target cell type, recovering it in an efficient manner is challenging with flow cytometry. It's also laborious and expensive. So one question that we had, since we know that ChBE technology enables high efficiency and high purity circulating tumor cell capture, is can we use that as a surrogate for flow cytometry so that we can perform single nucleus sequencing directly from the ChBE chip so that we can get this copy number variation information. And this is work that was done in collaboration with Jim Hicks back when he was still at Cold Spring Harbor using technology that was developed by Nick Nevin, who now has an independent laboratory. So I don't have time to go into the details, but I did a lot of confirmatory experiments to ensure that our ChBE copy number variation profiles were comparable to flow cytometry to enable all of our future comparisons. And I'm just giving you a flavor of the type of analyses that we did here. So this is, again, we're looking at the copy number profile for on average 150 cell nuclei. And now in comparison, we're looking at the copy number variation profile now from a single nucleus, a single cancer cell captured in our ChBE microdevice. And just qualitatively, by comparing the patterns of the profiles, you can see that they're almost identical. And you can, of course, quantitate this by directly comparing cell culture versus ChBE. You can see that we have almost identical agreement across these 2 different isolation pre-enrichment methods. Since we were able to confirm that cells that were isolated using ChBE technology did provide us with high resolution copy number profiles, we asked the second question of how does this compare to flow cytometry? How does this compare if we use, for example, 2 different cell lines from the same cancer type, but derived from different patients? Can we identify known and established mutations in each of the respective copy number profiles? And that's what I'm showing here. So this is one cell line type, LNCAP, again, isolated using the ChBE microdevice. And we're directly comparing this with another prostate cancer cell line called VCAP, which was isolated and amplified using flow cytometry, which is the state of the art. And in purple and red we have labeled different genes that we know are important for cancer progression that are well documented in both cell lines. And we're asking a simple question, can we tell the difference between them? So I'm going to annotate a couple of them just for ease of following by eye. One is looking at a pair of DNA damage genes that are differentially expressed in LNCAP versus VCAP. Another is a set of tumor suppressors that are differentially expressed, such as P10 and BNIP3L. As you can see, that there are differences in the copy number profile, which are validated by what we know based on the characterization of these cell lines. We can take a step back and do unsupervised hierarchical clustering of these cells to see if it discriminates by cell type or by cell enrichment method. The reason why we ask this question is if ChBE were having some sort of deleterious effect on the genomic profile of the cells that we're isolating, they should cluster separately than cells that were isolated using the current gold standards, such as flow cytometry. And the key takeaway from this is that cells did indeed cluster by cell type, not by enrichment method, indicating that our ChBE microdevices were not causing any sort of different changes in the cell genome. We're able to use this as a surrogate for flow cytometry to be able to investigate single cell genomic profiles with high accuracy and specificity. So to summarize this section, using rational microfluidic design, we can enable high purity isolation of circulating tumor cells for a variety of downstream analyses. We can adjust the ChBE microdevice in a variety of ways so that we can ensure efficient capture and isolation of the target cell of interest. And a simple example is us looking at genomic profiling, again, direct sequencing on chip, closing a technological gap that was existed previously in the field. So we just discussed how we can leverage microfluidics to isolate rare circulating tumor cells. This enabled us to address important questions such as patient response to chemotherapy and perform genetic analyses such as single cell cancer genotyping. In the next section, I want to discuss how we can further push microfluidic technology to identify circulating biomarkers at an even earlier stage of disease, early stage pancreatic cancer, rather than metastatic prostate cancer. The reason why this is a critical area, a critical technological gap that needs to be filled is because pancreatic cancer has really terrible survival outcomes. The 5-year survival rate is only 7%. It is predicted to be the leading cause of death by 2030. And one of the key reasons behind that is that cells appear to disseminate early in the disease development process. So even for patients where we think they have localized cancer and can undergo surgery for curative resections, most of those patients relapse within a few years. However, we have very few biomarkers for early detection and monitoring of pancreatic cancer. The lack of sensitive and specific biomarkers is what makes pancreatic cancer such an excellent target for liquid biopsy assay development. However, the key drawback is that circulating tumor DNA abundance in early stage pancreatic cancer is almost 6 times lower than what we find in metastatic cancer. So we've taken the needle in the haystack problem and made it 6 times worse. So to give you a general idea of what the ranges we're dealing with are, I've plotted a general idea in pancreatic cancer of what fraction of cell-free DNA you would expect to be tumor fragments in the various stages of disease. So for patients that have metastatic disease, you may have a circulating tumor DNA fraction that's up to 10%. And then as you would expect, it decreases precipitously the earlier in disease progression you get, to the point where at early detection, you're at 0.01% or even lower. Now, there are a variety of technologies that have been developed to try to address detecting rare mutant DNA fragments at a variety of allelic frequencies. Of course, Sanger sequencing is a field standard and something that we're all highly familiar with. However, in, I would say, the last 6 to 8 years, there have been a lot of next generation sequencing-based technologies that have started to push that limit of detection down to that range where we can perform early detection and monitoring. And this provides us with a lot of critical information to guide patient therapy in pancreatic cancer. However, despite the fact that we have increased the technical performance of these tools, our assay sensitivities are still extremely poor in pancreatic cancer. Our ability to detect circulating tumor DNA in disseminated disease, which has the highest tumor burden, is only 75%. And once you get to late localized disease or in a monitoring stage, you're basically at a coin flip of whether you're able to pick up circulating tumor DNA at all or not. Now, there are a variety of different technologies that are used to try to identify these circulating tumor DNA fragments that I outlined on the previous slide, and I want to briefly go over the pros and cons of each of them. So one technique that we're all very aware of and comfortable with is qPCR, which has been used for circulating tumor DNA. It has minimal hands-on time. It's a few hours to results. However, the drawbacks are that it requires high DNA input. You are getting indirect quantification. It has a really wide dynamic range. It's not really appropriate for early stage detection. And that's the reason why next generation sequencing has really become the gold standard, especially in pancreatic cancer. You're able to have absolute quantification. You have the ability to do de novo target discovery. You can explore the entire exome or entire genome. However, there are few key limitations that hinder our ability to implement NGS at a wide scale at a variety of clinical cancer centers. One of which is the hands-on time is significant, the technical expertise is high, the time to results are several days, and the amount of DNA you need to have is actually quite high. And remember, a couple of slides ago, I pointed out that the amount of DNA we have to work with in early stage pancreatic cancer is already 6 times lower than it was in metastatic. So for many patients, they just simply don't have enough sample to be able to undergo next generation sequencing. They need an alternative technology, an alternative strategy. And that's where digital PCR comes in. This iteration on qPCR uses the same reagents. The major difference is that you now partition your molecules into microfluidic droplets for the amplification step rather than doing it in a tube. This increases your sensitivity by several orders of magnitude. You can now do absolute counting because each molecule is in its own compartmentalized droplet. The hands-on time is minimal. The time to result is quick, and the DNA input is low. So if you have an a priori target, a list of targets that you know that you need to detect, ddPCR is actually a really attractive alternative. However, in pancreatic cancer specifically, assay sensitivity of digital PCR has lagged far behind that of next generation sequencing. So the question that I had in this particular project was, can we increase the assay and sensitivity of digital PCR while still retaining those input and speed advantages inherent to that technology? So the assay that I developed to address this technical gap, I call multiplex enrichment using droplet preamplification, or MEDAMP, and it's a spin on base digital PCR. So to review what digital PCR is, you're taking your original DNA sample, you're partitioning it into microfluidic droplets with normal target-specific fluorescent probes, usually TACMan. You amplify up your molecules in the droplet, and then you can do simple counting to detect wild type versus mutant, and that gives you a FACS style plot of frequency of mutant versus wild type. Where MEDAMP adds an extra layer of sensitivity is by adding an extra droplet preamplification step. So rather than directly partitioning our sample with probes, we instead partition our molecules into droplets with a high fidelity polymerase set. We amplify up that DNA so that we get uniform increase in signal of both mutant and wild type. We do normal DNA purification and then repartition for downstream ddPCR, digital PCR readout. Now, this is technology that I worked on in collaboration with 2 of my colleagues while I was at MD Anderson. One was Dave Zen, who's a medical fellow who was really instrumental in gaining the clinical samples that we used, and also Rob Cowan, who worked really hard on developing the first set of primers we used for this device, for this assay. So one of the first questions was baseline validation. How does this perform with controlled genomic standards at allelic frequencies that we understand? Is this strategy of pre-amplifying your DNA in droplets more sensitive than just pre-amplifying the DNA in a tube? That's our first key question. And that's what these set of digital PCR plots show. So on the y-axis, we have VIC, which is our wild type fluorescent signal. On the x-axis, we have FAM, which is our mutant fluorescent signal. For a sample that had 0.13% of tumor DNA, you can see that the wild mutant signal is quite low. You only have 4 positive droplets. That is not robust enough to inform a clinical decision in the hospital. You need a more robust signal than this to make a clinical decision. In contrast, if you do PCR preamplification just in the tube without using our MEDAMP strategy, you vastly underestimate the allelic frequency of the tumor DNA. So you're underestimating the tumor burden that the patient has, and that could have great consequences in terms of what therapy that they should pursue or whether their cancer is under control. However, if you use the MEDAMP strategy, you now see that our allelic frequency is accurate, we retain that information, and we now have a very strong and robust mutant signal. And this is the case across a wide range of allelic frequencies, ranging from about 0.02% to up to 10%, which recapitulates a physiological range of ctDNA allelic frequencies. Once we validated that the MEDAMP strategy worked using controlled synthetic DNA samples, we then looked at metastatic patient data. And an example of that is shown here. So we have a benign or normal sample that should have no mutant DNA in it. And you can see that the signal, as expected, is quite low compared to a metastatic pancreatic cancer patient, where you can see that the mutant signal is almost to complete saturation. And I want to point out that the amount of DNA that we're using in these assays is quite low. You're using under 3 nanograms of DNA. This is really pushing the limit of what we can do with digital PCR or next generation sequencing, period. So we're really starting to, again, get at those samples that would otherwise be completely disqualified for molecular profiling. I summarize this data here, where we're again looking at the set of benigns that we tested versus pancreatic cancer, which is labeled as PDAC. And you can see that a couple of the benigns are actually KRAS positive for the mutation of interest. Both of those patients actually had colorectal polyps, which is kind of an interesting point, which I'm happy to talk about later if people have questions about that. Now that we established that the MEDAMP technology is extremely sensitive and now reaches that threshold that I mentioned to you earlier, we wanted to get to the 0.01% level. We reached the 0.02% allelic frequency level. We now extended this technology to look at a cohort of early stage pancreatic cancer patients. So we had a set of 71 pancreatic cancer patients that had varying stages of pre-metastatic disease, and then 20 patients that were metastatic as a control. So one of the first questions that we had was, how is MEDAMP performance in those low DNA input samples now that we have those pre-metastatic clinical samples? They're going to have a lot more biological heterogeneity. And the answer is that MEDAMP performed quite robustly across the range of DNA inputs that we tested. So I'm focusing on the low DNA input samples, which I'm defining as 20 nanograms or lower. And you can see that the percent detected is uniform across this range. And if we compare this to what was at the time the state of the art, next generation sequencing, as I pointed out a few slides ago, sensitivity in pre-metastatic patients is at about 55, 60-ish%. Normal digital PCR is quite abysmal at about 38, 40%. However, if you add the simple digital preamplification step, you now push your assay sensitivity to be in line with this gold standard, but with much less turnaround time, much less labor, much less technical expertise required. And this is about a 2x improvement in sensitivity with minimal additional time in your workflow. Now that we have a tool that's able to reliably detect circulating tumor DNA in early stage pancreatic cancer, we can now start to ask fundamental questions like, does circulating tumor DNA burden correlate with overall survival, which is what I'm showing in this Kaplan-Meier curve, where again, we have overall survival probability on the y-axis, and we're tracking those patients over time in the x-axis. And you can see patients that have a lot of circulating tumor DNA have much less have a much worse prognosis than patients with low circulating tumor DNA. For the small sample, for a small cohort of patients, we actually have matched tumor ground truth specimens for whether they had this mutation or not. And I'm showing a couple of examples of that here, where we have a patient that we know had a mutation in the codon that we're looking at in plasma, and we see that same mutation in the circulating tumor DNA. This is a patient where we know that their tumor did not have a mutation in the codon that we're checking, and as expected, we have no signal in that patient that had a KRAS negative tumor. So in summary, this technology enables robust detection at allelic frequencies as low as 0.02%. And why is this important is that it now allows us to look at that earliest stage of pancreatic cancer progression. It allows us to detect biomarkers in patients that would otherwise be discarded for having too little DNA of insufficient quality. I would like to emphasize that this is just a molecular assay test. We're not looking at clinical history. We're not looking at any other complementary clinical biomarker data. Just this one single gene with four targets is enough to give us this level of resolution and sensitivity. And that allows us to ask fundamental questions about pancreatic cancer biology that would otherwise be extremely challenging using standard technologies such as digital PCR. So in my talk, I covered how we can use microfluidics to enable us to capture circulating tumor cells, that we can ask questions about chemoresistance, so that we can ask questions about individual cell genomic profiles. I also showed you how we can now use droplet microfluidics to look at circulating tumor DNA to ask questions about molecular profiles in the earliest stages of pancreatic cancer development. In the last few minutes of my talk, I want to briefly go over what I'm working on in my current postdoctoral laboratory, where we're developing proteomic tools. And now this allows us to ask functional assay information about the cells rather than just looking at the genome. So in my current lab, we focus on protein kinases, which regulate key oncogenic signaling pathways, but we have very few tools and very few inhibitors to address them. So of the 500 kinases in the human kinome, about 350 of them we know are associated with cancer pathways. However, we only have 62 FDA-approved kinase inhibitors targeting about 24 of those kinases. The reason why we have so few FDA-approved inhibitors is because only 5% of kinase inhibitors that are in development actually have any clinical utility. Only 5%. The reason why this turnaround is so poor is because the current method for testing kinase inhibitors in a high throughput manner is to test a kinase outside of the cell. It's a kinase in a tube with a peptide substrate, and you add the inhibitor and you measure how active the kinase is on that substrate or not. However, we know that substrate sequence only explains 25% of in vivo kinase specificity. Kinases are highly spatially and context specific. They rely on the regulatory machinery around them to control what substrates they see when, and that dictates their specificity. When they're tested outside in a petri dish, for example, they actually will phosphorylate things quite promiscuously and give you false information about whether your inhibitor is effective or not. And of course, there's a reason why we focus on biochemical assays for inhibitor development, and that's because current cell-based assays are extremely challenging to implement in clinical specimens. And this is a brief overview of some of the types of assays that you can try to implement in a cell-based manner. So if you know a direct substrate of the kinase, you can try to detect that directly with phosphospecific antibodies, but that requires a priori knowledge of a target. You have to develop a phospho antibody that's specific to that substrate, which can be quite difficult, and it might not be sensitive enough. There are also assays that look at intermediate steps, such as looking at downstream substrates that you can genetically encode into a cell to give a fluorescent, such as a FRET reporter. However, you can't genetically encode a reporter into a clinical specimen, obviously. You can look at more distal assays, such as proliferation or functional metrics, but that doesn't really give you information about what that kinase specifically is doing. That's giving you a bulk general outcome. So in the Parker Laboratory, our strategy to try to address this need for a cell-based measurement that is easy and can be implemented in a high throughput manner is to use artificial peptide probes. And that's broken down into several compartments, one of which is the enzyme substrate. So this is a series of amino acids and a motif that we know that the kinase preferentially prefers to phosphorylate compared to other substrate sequences. We combine that with a bioconjugation handle, which can be as simple as an amino acid that contains a biotin group so that we can perform an ELISA-style sandwich assay, or it can be something like a reactive cysteine so that we can perform thiamalumide chemistries to attach a fluorophore. The final module of our cell deliverable peptides is a cell-penetrating peptide, which is a sequence of amino acids that through a variety of highly contested mechanisms is able to locally disrupt the cell membrane and traffic cargo into the cell. So this combination of modules, the enzyme substrate for the kinase, the bioconjugation handle for detection, and the cell-penetrating peptide for delivery, is what we call an artificial peptide probe. This can then be simply incubated with your cells of interest, where it will be trafficked into the cell, and then the endogenous kinase within its native context with its regulatory network can phosphorylate or not phosphorylate that target. Thanks to the bioconjugation handle, we have a variety of different tools and methods and readout strategies to detect kinase activity using these artificial peptide probes. So one of the key questions and limitations towards designing an artificial peptide probe is picking one that's clinically relevant. So in our laboratory, we have a variety of different methods where we try to have a kinase-driven hypothesis that informs our identification of a substrate sequence that's specific to that kinase. We look at binding ligand partners if possible. We do in-cell and in vitro validation of the specificity and catalytic efficiency of these artificial peptides. And then depending on the bioconjugation strategy that we employ, there are a variety of readouts that we can use, ranging from mass spectrometry to imaging to fluorescence. And because these modular peptides are so flexible, we can use them in basic research where we're trying to understand kinase inhibitor efficacy outside of the cell, and also in translational contexts where we want to look at cell-based kinase endogenous phosphorylation activity. In the last couple of slides, I'm going to show you a couple of examples how we can use these artificial peptide probes to, again, look at endogenous phosphorylation activity in a cell-based manner in a physiologically relevant context. So one example is looking at cell lines that have been incubated with an artificial peptide probe that's specific to two different kinases of interest, one of which is Circ, one of which is Able. Our peptide probes are labeled with two different fluorophores, one of which is dilate 488, one is dilate 550. And this is the same set of cells labeled with two different artificial peptide probes with two different fluorophores. So this is a multiplexed readout of Sarc and Able activities simultaneously. And these cells, if we perform EGF stimulation, we see a net increase in overall signal. If we add imetinib, which is specific for Able, you'd see a decrease in overall signal from the Able peptide probe, but not from the Sarc peptide probe, showing that these peptides are truly kinase specific and can give you real-time information about kinase signaling pathways. You can add an extra layer of complexity to this by starting to monitor kinase response to therapy in a time-dependent longitudinal manner, and that's what I'm showing in this graph. So again, these are the same set of cells. We're following them over the course of minutes, so from 5 minutes to 40 minutes. We have EGF stimulation at the top, and then we have the dasatinib treatment at the bottom. And the key readout here is to look at the relative difference in fluorescent intensity over time so that we now start to get a curve of phosphorylation activity. So you see with EGF stimulation up to 40 minutes, we have an increase in phosphorylation activity until it reaches a net peak. We then introduce the kinase inhibitor and we see a precipitous decrease in phosphorylation activity, and we can read this all out again using our artificial peptide probe rather than more laborious endogenous substrate detection methods. So my current work focuses on integrating bioinformatics with peptide engineering so that we can continue to design clinically relevant probes that has two different thrusts, one of which is how we select our targets. I do that by integrating biorepository data such as TCGA with identification of high-value target kinases indicated by the FDA so that we can find ones that are differentially expressed in cancer versus normals, for example, and also ones that correlate with overall survival probability so that we can pick kinase pathways that might have the highest clinical impact as quickly as possible. The second wet lab component to my research is substrate design, where I look at different cell penetrating peptide strategies, characterizing their uptake and loading into cells, and then implementing them into different physiological contexts. And an example of that is our investigating kinase activity in these 3D collagen microsphere constructs, which is an ongoing collaboration with Dave Wood at University of Minnesota. So over the course of my talk, I've covered 3 different strategies that we've used to try to close the gap between the possibility of liquid biopsy and the actual ability to implement this to answer key cancer biology questions, whether that's looking at molecular genotyping, whether that's asking questions about cancer progression and dynamics, or whether that's looking at functional activity. And I'll briefly discuss how I plan to implement all 3 components of this in my research program as I move forward in my independent research position. So again, my laboratory is going to focus on integrating engineering with multi-omics so that we can develop these non-invasive tools. I'm specifically interested in the diagnosis and monitoring stages, especially longitudinal monitoring. My research program will have 2 key thrusts, one of which is a clinical thrust looking at molecular profiling and molecular screening, extending that mid-amp technology to encompass things like chemoresistance profiling over time. And also a preclinical thrust where we're looking at functional analyses and how we can integrate those with circulating tumor cells. So now we can have a functional biomarker assay for liquid biopsy, which is currently something that is critically lacking in the field. The overall long-term vision of my research program is to integrate both of these so that you can get proteogenomic information and a microfluidic system so that we can perform single-cell multi-omic profiling with the idea that the complementary information provided by molecular profiling and functional profiling can help us pick better drugs and better treatment strategies for patients. So I want to conclude by acknowledging all of the people that I worked with extensively over the course of my graduate and postdoctoral career. So Parker Laboratories and Preeti Jenna worked a lot on the peptide probe work that I cited. RIM Lab, again, Rob and Dave were really critical for getting MedAmp up and running. My clinical collaborators, without whom this would not have been possible over the course of my career, and of course, other collaborators at Cold Spring Harbor Laboratory for sequencing, and my funding. So thank you. I'll be happy to answer any questions.
Well, thank you. It was a great talk. Like a silent clap. So we have some time for people who have questions. So I'm going to open it up to the audience, and I just, if you have a question, just feel free to ask.
I have a question. That was a beautiful talk, Erica. Thank you so much. My question relates to your JEDI technology. I was really impressed by your ability to detect, I think you said 200 to 400 times more CTCs than the clinical standard, which is Veridex. I'm just curious, what was your definition for a CTC? I think part of the reason why they're sort of stringent is because, it has to be CD45 negative, EpCAM positive, and so on. So what sort of criteria did you use?
Yes, that's a great question. That is an inherent catch-22. I will admit in liquid biopsy, we beat up on cell search a little bit due to their, as you said, they have to be extremely stringent, both in terms of surface marker expression, nuclear to cytoplasm ratio, and cell size to call a CTC a CTC. And that was so that they could get FDA approval. So yes, their definition of a circulating tumor cell is extremely stringent compared to what is used in the field. But since it's the only FDA approved standard, we still compare against it. Our definition of a CTC is EpCAM positive, CD45 negative, which is fairly standard within the CTC research field. We also added on the layer of being PSMA positive for the research that I presented, because we also focused on prostate cancer specifically, where there's a really nice antibody for that prostate-specific membrane antigen, which we have some data to show was robust to epithelial to mesenchymal transition, for example, which is why that made it such a nice immunocapture marker. But yes, there have been, I don't know if Veridex has ever showed it publicly, but they have shown at like Gordon conferences and things like that, if they relax the criteria to what the field standard is, the amount of recovery is significantly higher than what is reported officially. You're absolutely correct.
Thank you.
Are there other questions?
Erica?
Yes.
So in your droplet amplification step, so it's a water and oil emulsion. You're kind of dividing your sample up into different emulsions. Is there an issue with, first of all, are you adding endogenous surfactant, or are you relying on just kind of the the blood proteins to stabilize that interface? And do you see any kind of adhesion of circulating DNA to the interface because it's a fairly large amplifier?
Those are great questions. So for the technology that I presented today, all of this was developed on Raindance. So it was a startup technology company that was recently bought out by Bio-Rad. So yes, they do have surfactants, which are patented information to help stabilize their microfluidic droplets. It is not as robust as the Bio-Rad system, since they have that protein hardening function, which helps with containing your molecule after amplification. Raindance does not have that technology, did not have that technology prior to being bought out by Bio-Rad. Does that address your question?
Part of it, and then is there, even though if you have surfactants, you can also get displacement by biomolecules in the solution. Did you see any evidence that, for example, you're getting, marker fluorescence from the interface of the droplets or anything like that?
We didn't see that, but that's because there's always an upstream cleanup step. So we're never running sample directly unpurified. For the data that I showed, this is circulating tumor DNA that's been isolated directly from plasma. So there's a lot less autofluorescent molecules that could interfere with the reading. There are certainly small amounts of PCR inhibitors that still make it through, and we do see inhibition of amplification in those samples compared to, for example, just pure TE or PBS or something, water. But no, we don't see issues with autofluorescence, but that's probably because of the upstream cleanup.
Thank you.
Just to follow up on that, for somebody who doesn't know much about ctDNA, is that the only DNA you're capturing from plasma? I mean, or is that just the only DNA you're amplifying, basically?
So for the assay that I presented, we're amplifying both. So we have two sets of primers, one set of primer that's to the wild type version, and one set of primers that's specific to the mutant version, so that we can get relative abundance information. So we get both prevalence of background wild type signal and also our mutant signal. And that's how we're able to report those variant allelic frequencies that I mentioned in my talk. There's certainly other strategies that focus on trying to degrade away the wild type and simply detect the mutant, which is an easier and more straightforward way to increase your signal to noise ratio. But then you lose that allelic frequency information. So you lose the tumor burden information, what I think is just as important as whether the circulating tumor DNA is there or not there at all.
Okay, thanks. Are there any other questions from the audience? I haven't heard any grad students asking.
I've got a question. So I was wondering if from your sequencing data, if you can, or just from, looking at the genome of these cancer cells that have extravasated into the blood vessels, I'm wondering if there's any sort of genetic expression shift that could tell you how advanced a cancer form is, or if that's just way off the mark with this kind of work.
So that's absolutely an active area of research, is trying to draw the phylogenetic tree between what you see in circulation, what you see in the primary tumor, what you see in a metastatic tumor, so that you can interpolate like what stage is the circulating tumor cell in as it pushes its way through, and what the hope that maybe you can find some subset of alterations that help a cell make its way into the bloodstream. It's still an active area of argument whether they are making their way through leaky vasculature and making a escape of opportunity, or whether they're actively degrading their nearby ECM to make their way through. That's something that's still being actively debated in the field. There's some mouse model data to suggest that there is active tunneling being done to push their way into the bloodstream and see the second metastatic niche. So that is a great question, and absolutely something that's being focused on. As technology gets better and better, I'm sure we'll be able to address those types of questions.
That's awesome. Thank you.
Anybody else? Okay, so with that then, I would definitely like to thank Erica. That was a fantastic talk. And
Thanks for having me.
It's clapping, but we are clapping. And with that, and if anybody wants to stay behind, we have about 15 minutes before her next meeting. So with that, thank you, everybody.
Thank you.
And I think